invitation to come and speak at this Science Week at the UM6P. It's a pleasure to come back here. I was here a few years ago and come back to Morocco. First time I was here was 1978, which was a long time ago. And also meet again old friend from college days, Ghani. Really appreciate being here. So I'm going to talk about under the theme of complexity, about climate change and specifically the role of water in the soil, plant, atmospheric continuum. And I'll explain why in a second. First, I posit that global change, as we define it today, is a response of a very complex system to perturbations. It's a very complex system because it touches on almost every branch of investigation, scientific investigation we know. It involves physics because there's ocean currents and winds that transport heat and energy and create weather, so it involves physics. It involves chemistry. There's biogeochemical cycles, carbon, methane, nitrous oxide, all sorts of oxygen that are involved. And not only involves chemistry, but it involves all aspects of chemistry. It involves solutions chemistry, like chemistry in gases and in aquatic chemistry in the oceans, but also heterogeneous chemistry, particulates, aerosols, soils with surfaces, very complex branches of chemistry. It involves biology because much of the chemical cycling is through phytoplankton and to plants, and they don't have necessarily governing equations. And also global change involves the sort of the human dimension. It involves both sort of the rational branches of human behavior, like microeconomics, macroeconomics, how systems adjust in a sort of with governing laws, but also irrational aspects like behavioral economics. We do things that not often makes economic sense, or cultural politics, that they don't have governing equations and they don't conform to any laws or predictive laws. So it involves all of these in one sort of a complex system. Now, what do I mean by perturbation? So that was just a definition of what the global system is. The first perturbation that we all hear about all the time is the process of taking carbon that's locked in coal, oil, or gas underneath the ground, so it's sequestered, and we mobilize it and put it in the atmosphere by burning fossil fuels. So this is a record of carbon dioxide concentrations in years at an observatory in the middle of the Pacific, nothing upwind, nothing downwind, and it keeps going up because of you're taking stuff that was locked up and re-releasing it in the atmosphere. This absorbed radiation and hence climate change that we know it. But there's another global change that has been going on that has already happened. It's in the past and current. It's been going on for decades. And that's when we, for instance, deforest or change grasslands and savannas into agriculture. You fly over almost anywhere and what you see is a changed landscape. The impact of that to date, as far as the water is concerned, is far larger than this could possibly be. For instance, evapotranspiration that Ghani talked about, or the recharge to the aquifers, the natural recharge to the aquifers that we pump water out of, they may have changed because of these by a factor of 2. Is not surprising to see. They have either doubled evaporation or doubled recharge, or halved it, reduced it by half. So this is a water cycle global change that has already happened And it's happening at a much larger rate than this. Now, so this is a very complex problem. Our perturbations are pervasive and major in magnitude. So how do we get our arms around this and deal with it? So what I'm going to basically start is look at the biggest metabolic cycles of this system. The three main metabolic cycles that determine our climate is water, energy in terms of solar radiation and thermal radiation back out, and carbon. So let's just consider for now the three main cycles that maintain the climate system. And as Gani mentioned, and in our domain and in this session, what we're interested in is how perturbations in any one of these three cycles eventually affects the water cycle. So we want to look at all three cycles, but what does it mean for changes in the water cycle? As Gani mentioned, for instance, the extremes or even average precipitation is changing. So that's the ultimate question. So what goes into making predictions about these three cycles and global change? We built climate models, numerical models of climate, and run them forward for 50, 100 years to see what the average response of the system is to these perturbations. So a typical climate model would start with the physics. So it would take the entire globe, divide it into nodes in horizontal and vertical dimensions, and then solve equations so that they're connected with one another. For instance, we solve for conservation of momentum, which is physics, conservation of mass, it's a physical constraint, thermodynamic equation. So we have enough equations for each and enough unknowns to solve the system. This is a very expensive computational problem. Only a few centers can do it. So a few centers around the world do it, consortia usually. But also this is now has become, it's such a heavy computational problem, it has become the standard for benchmarking computers. So when companies come up with new processors, they use these to benchmark how fast the new system is working. But that's only physics. What about all the other chemistry and biology that goes into it? So not only you have to have that, but you have to include chemistry and biology and also emissions by humans all into it. I will look at one particular example of what else we need to put besides the physics, which is the biology and the chemistry. And if you look at the surface leaf of a plant under a microscope, the leaf will have openings like that on it, stomata, and these things, the carbon dioxide and the water get exchanged through these tiny little holes on top of plants. Now, plants have, over evolution, have adapted to be able to basically inflate these cells at the surface of the leaf to close and open up depending on stress. If there's water stress, heat stress, nutrient stress, whatever stress there is, plants, in a sense, regulate how much they uptake carbon and how much carbon they release. Each plant has over 1,000 millions of years adapted to respond differently to different stresses. The unfortunate thing is that I can't ask a plant what rules they use to open and close that stomata so I can program it into the computer. So we can't ask that question. We have to somehow observe it. And remember that because I'm going to come back to that. Now, this is just one example. There's microbes working in the oceans and in soils, so the complexity of those as well. But this is, in a sense, a feedback control system that is not intelligent, but it regulates and responds to stress. So now, what are the sources of uncertainty? Well, one major one is that I can't ask a plant how they behave or respond to stress. And if you think of it together, that uncertainty propagates through the system. The three main metabolic cycles are like gears in a clock, that if you perturb one, you will see perturbation in the other, even though you didn't directly perturb it. The question is, how are they connected? How does change in one respond or correspond to another? The problem is that, like the plants and the microbes as I showed, not so much the physics, we have to have relationship defined for those that we don't know, so we guess. And if they're wrong, then perturbations that are put into the system propagate differently across. And this is a major source of uncertainty in global change predictions. And that's why, like Gani mentioned, they become unreliable for decision makers. They're not willing to turn the economy around just because one model says this and another model says another. So let me basically call that statement of the problem. That is the problem. This is, for instance, how evaporation, that Gani talked about, responds to soil moisture. So on the x-axis is soil moisture, and evaporation will obviously go down when the soil becomes dry. So when you go on the low end of soil moisture on the x-axis, evaporation goes down. Now, this is the response from, 6 climate models. 6 different high-performance computing centers. Canadian one, French one, German one, Norwegian one, doesn't matter. 6 high-performance centers that create these climate models. And this is how they model how the cycles are connected between evaporation and soil moisture. They don't even overlap. The 3 main metabolic cycles, I can't get even the basic one agree among the models. It's because we used all different imagination of what plants do when they get stressed and so forth. So that is the problem we need to tackle. And if you look at carbon flux, grams of carbon per day, flux per meter squared, versus soil moisture, this is how plants take up carbon or close their stomata over a particular region. It doesn't matter where. It's just 6 different models. It's all over the place. This is not solving a complex problem properly. So that is the problem that we're trying to solve. The way to solve it is by observations, and global observations, so it covers all sorts of plants, all sorts of climates, all sorts of conditions. So in order to bring these models to harmony, what we need is pure observations of the x-axis and the y-axis, just based on observations, so that you have a benchmark and say, model A, B, and C, you should be on this line. You can't be anywhere you want. So that's remote sensing. You have to observe each of these separately. That means you have to use many different sensors, because each sensor gives you a different piece of information. So this is the exercise that I'm going to walk through, starting with soil moisture, the x-axis. This is a satellite mission that I've been working on for 20-odd years, and now it's making measurements. It's a NASA mission. The name is Soil Moisture Active Passive, so it measures soil moisture. We have evaporation, direct observations, and independent from weather stations across the US, diverse climates, pretty good station coverage. And then for carbon flux, there are US and European missions that measure fluorescence. Fluorescence is the energy that's put out during the process of photosynthesis. You absorb solar radiation, you use a lot of it for photosynthesis, you release some of it as dissipative heat, and some of it is shifted in frequency and re-emitted. So it's an emission above natural background, and it's direct measure of photosynthesis. So you have carbon flux, water flux, and the soil moisture. So put that all together. This is what I would call probably the most expensive plot there is. 2 satellite missions, terabytes of data in one plot. So that's the same graph as we saw before for the climate models. SMAP soil moisture on the x-axis, evaporative flux normalized, so it's between 0 and 1 on the y-axis. We did this over the US, so there's different types of vegetation, like arid Southwest, Midwest agriculture and forests and so forth. So we see that you see the same behavior, but now with only observations. And a climate model should be on this curve. So it's a benchmark. And the same thing for carbon flux. Now we have solar-induced fluorescence versus soil moisture. The units doesn't matter. We want to interested in the shape. It has the same behavior. When soil moisture is low, the plant and the evaporation are low. When the soil moisture becomes higher, it increases, but then it reaches a plateau. There's plenty of water, but not enough energy, or not enough nutrients, or anything else. So when it's just evaporation, it plateaus to the same number. With different vegetation types, it plateaus to a different number. Different vegetation have different sort of maximum capacity to uptake carbon. And does this match what we expect? This is all from satellite, so you're not touching anything. You're just observing images. Now, if I look at water use efficiency, which is the division of these 2 axes, and I'll explain what it is. Water use efficiency is how much carbon I take in per unit water vapor that I release. So photosynthesis is carbon in, water out. That ratio of what is the exchange rate between those depends on the plant type, and also it depends on stress on the plant type. When you plot it this way, the plants have a certain efficiency, but then when they get stressed at low soil moisture, they start regulating, like we talked about before. They become much more efficient in using carbon, but at an expense. They get damaged if they do it for too long. It's a very strong adaptation, so when you're stressed, you become much more efficient as a plant. Now, you can do this in chambers, in a laboratory, and ecologists have been doing it, agronomists have been doing it forever. They put a plant in a chamber, they look at water use efficiency, you can measure water vapor coming in and out, and then they stress the plant artificially in a laboratory. They torture it, basically. So you get the same behavior. So you're seeing from satellite what you would see in a chamber, but now not for an individual plant, but globally for all sorts of plants. So let's move on to look at variability, is what Gani talked about, extremes. What causes variability in available water resources? Evaporation, recharge to the groundwater aquifer. What is the cause of the year-to-year variability in that? It would first guess, I would make, and I think it's reasonable, is that if I look at variability in precipitation, I'm just going to see that variability in evaporation. The more variable the input, the more variable the output. Is that really the case? So this is variability of precipitation interannual across the continent, so millimeters per year. If I look at the variability in the response of the system, for instance, if I look at variability in the range in temperature from morning to evening, how fast the system warms, it depends on the moisture. On a dry day, it's a lot more. On a moist day, a lot less. The patterns don't even match. So there's more to it than the forcing. What is that? Okay, we have, again, satellites. So if I can answer that, I can answer where is it most vulnerable to climate change? Which landscape, where is most vulnerable to climate change? And what drives the difference between these two maps? What's in between that makes it not a one-to-one map? So we go back to this figure here. What this figure shows is that there must be two regimes going on. There's a regime where the soil is quite wet and the evaporation is just not responding to soil moisture. There's plenty of soil moisture, but not enough energy, not enough sunlight. And then there's a threshold in which there's enough energy, but not enough water. So evaporation responds very strongly. There's a slope between evaporation and soil moisture. So there's two regimes, and you switch from one to another between storms. There's the energy limited and the water limited regimes. Two entirely different physics. Not a continuous physics, but physics that changes rules when you go from one to another. Well, when does that happen? How dry should it be for that to happen? So again, two regimes. This is the red surface temperature. You see that when you go across a certain threshold, the temperature responds. It doesn't respond up to that point. And this is sort of direct measurements of evaporation from very expensive tower flux instrument towers that you can put. And it has that evaporation behavior. So temperature range is a proxy for evaporation. And temperature you can remotely sense. Evaporation you can't. And this is using now combining two satellites, SPAP and the MSG Meteosat second generation geostationary satellite that's sitting actually on the equator across Africa. So the best data is actually over Africa, not for weather forecasting in Europe. And we're going to look at a few regions as an example that are color-coded. And you see the temperature responses over them. So it's a pretty good proxy. And in the sort of process of analysis and all that, we figure out those soil moisture thresholds. This is the threshold that separates water-limited and energy-limited regimes and how they're distributed globally. That lower threshold is when plants just stop. That's the wilting point. They just can't go below that. And then this is the drainage to the recharging the aquifers. So three different thresholds, three different regimes. And this is the fraction of time that you're in for one of those regimes. And the other, it says 0 and 1, so in the other fraction in the other regime. And this is the soil moisture and the evaporation regime. This is a threshold. Now, it makes a big difference if the soil moisture is distributed here or during anomalous years it shifts. The response will depend where is this distribution relative to this threshold. And when you do that, you can then map landscapes that are particularly vulnerable. That means that where they are with their distribution of soil moisture is right near the threshold. So these regions are particularly vulnerable to a little change in precipitation variability. And this explains why the regions with the most variable precipitation are not necessarily the ones with the biggest evaporation or drainage or recharge variability, because this is the filter through which they go through, and this nonlinear filter. Okay, I'm going to end, if I have 5 more minutes, okay? Yeah. End with what we talked about at the beginning. I cannot ask a plant to tell me what rules they use to shut down their stomata and reduce evaporation, what adaptation do have to water stress, to heat stress, and so forth. So we can't ask them, but if we take a lot of pictures of them during various states of stress, can we figure out what is their internal sort of rules without knowing? So again, we go back to soil moisture because that's where plants get their water. But now we need to know how happy the plant is. So for that, it turns out that at least with the SMAP machine, we have an in-house algorithm, not the official algorithm. In the same way as we measure soil moisture, we can also estimate in the process from the same measurements the moisture in plants. So it's the same variable, instead in the soil, how much water is stored in the plant. And then we can detect how much water the plant is losing under different soil moisture conditions. That's the title of the talk, Water Movement in the Soil Plant Continuum. Basically, what we're trying to do is to figure out the rules that plants use for responding to water stress by just taking a lot of pictures under many different conditions. So how is that done? Just as an item of interest, example, the red line, the blue line are soil moisture. They go up and down, rain events. For instance, this red dots identify one particular dry down. So soil moisture went up because of rain, and then it slowly goes down until the next rain, it shoots up again. The green line is the amount of water stored in the plant. This is the response of the plant to water availability. So let's just take a few dry downs, a few events. So these are just a few examples from the long time series. The soil moisture in blue is always going down because by definition I picked periods where soil moisture is drying down after a rain event. So it goes down, the blue goes down in every one of these. What is the plant response? In the first one, the plant also goes down, and then it goes back up. Why? In the second one and third one, it always goes up. In this one, it goes down, and then it goes up again, even though there's no rain. Okay? So this gives us a clue that the plant is not linearly responding to soil moisture, but depending on where the starting point of soil moisture is, what the temperature environment is, there's some other factor that mediates how the plant responds to water stress. So in order to visualize that, instead of looking at things in time, which makes it impossible to figure out what's going on, I eliminate time between these two time series. So I make a phase space. How do these two variables evolve together? So instead of time, I plot the soil moisture versus the water, vegetation water content. So this is one of these events. It starts wet after a storm, and it dries down, so it's going from right to left, and the VWC in this case is going up. But sometimes it goes down. There's not much to see here, But if with remote sensing, I don't have just a few dry downs at a location. For each one of these, and over time, I have thousands of these lines. So, for instance, for one of these, 100,000 pictures of the plant responding. So now I need to clean this up to sort of distill what is the plant behavior, that rules that it uses, not intelligently, but evolved over time with adaptation. So if we just, instead of putting them all together, I bin them and look at evolution from every starting point. It looks something that I can visually, it's the same graph, it's just cleaned up. So when a soil is wet after a storm and it dries, the plant uptakes water in this particular plant type, and vegetation grows in water content, and then there's a threshold in which it crashes. It just can't withstand it. So this is the behavior of that plant that we couldn't ask it, but now we can see it. And this is going to be a different number, and this slope is going to be a different number for each plant type. So here's, for example, 2 different plant types. This one's definitely a different behavior. And we plot the slopes, we plot the thresholds, and basically try to reconstruct the plant response behavior to stress. And then let me just, this is the last slide. If I look at any one event, it looks like this. So there's a precipitation, and then the clock begins on dry downs until the next precipitation event. So day 0, day 1, day 2, so that's the precipitation event. Soil moisture jumps up as a result of precipitation, and then it goes down. If I look at those vegetation water content, they kind of look like this. They have a delayed peak. Now, the question is, going back to the cycles, how much of this is rehydration? The plant is dry. Soil moisture goes up, and the plant, like a sponge, just picks up the water. That's one reason why this could happen with a delay. Another reason is the plant is actually growing. It's growing new leaves. So we need to figure out, again, a behavior. How much of it is rehydration? How much of it is actual plant growth or crop growth? The difference is that in one of them, there's no carbon exchange. It's just sponge. In another one, you're taking in carbon. So that's, again, trying to ask a question from a plant without it knowing your language. So we look at changes in carbon, in above ground biomass for different attributes that the plant must have figured out to respond to. So what are the environmental conditions in which you just act as a sponge or you actually grow leaves? So in conclusions, the global change problem and future sustainability of human welfare involves doing these simulations of the global system involving physics, chemistry, heterogeneous and solution chemistry, biology, rational human behavior, and irrational human behavior, all in one. So this is, how much more complex can you get? For some of those domains, like heterogeneous chemistry and human behavior, you don't even have governing equations. And for some of it, like plants and microbes, you have to come up with what evolution has taught those plants and animals. The first step is what I was trying to talk about, is by observing the system and using natural variability of precipitation variability, storms and interstorms and all that, as small perturbations, and observe how they respond to those perturbations, so that then I can perturb them on a much larger scale. And we can't just assume these responses. We have to observe them and then code those observations. So, like I was trying to do, was to observe the x-axis and the y-axis, no models, and then create benchmark data sets that the models must conform to. And we're making plenty of these observations. Terabytes of data are being produced every day with observing systems. But you can't interpret them, just too much. And data science, machine learning, artificial intelligence are a big help, but they have to be physics informed. You have to provide them with what questions you're trying to answer. Machine learning and artificial intelligence won't both ask questions and answer them. You have to pose the questions, and that's physics informed. And those terabytes of data are sitting on the floor for the most part, and that's the conclusion, the last bullet point that Ghani had, and I have the same thing here. We need the persistent resolve of agencies and companies to make use of this new technologies, and the workforce, the most important part, to use these observations. Okay. Thank you.
That's okay. It was a very interesting. We can take a couple of questions.
Thank you, sir, for
Present yourself.
Hello, I want to thank you for your time and for the presentation. My name is Ayub Kishore. I'm a PhD student in the URI lab. So my question, you have talked a little about solar induced fluorescence. So my first question is about this, and how can we use it to monitor plant stress in terms of water? How can we use solar induced fluorescence to monitor vegetation stress, vegetation response to water stress? Is there any work that has been done because I'm seeing that people now trying to use it with thermal infrared parts of satellite products that use thermal infrared to monitor stress. Have you ever seen some work working on this? The second question about, I'm seeing that your team working on the vegetation water contents using SMAP. I think that you are using the L-band. Did you test using X or C band to monitor water stress? Because I know that L-band with the big penetration will give soil moisture, so I think you're using soil moisture in the soil with the water in the atmosphere, and the stomata response to the water circulation, to have an idea about vegetation water contents. But how about using some microwave satellites that have low penetration and also sensible to dielectric constants?
So
thank you.
Why don't you take the SIF question? I'll do the microwave.
No, basically, I mean, the SIF is the aerial tools to monitor how the vegetation, is dealing with water stress. I mean, as Dara presented, because the SIF basically is a direct measurement of the functioning of the vegetation, of the photosynthesis and carbon, etc. So, a plant that it is stressed in general, it is stressed, so it doesn't assimilate any carbon. While it is where it is not stressed, So there is a normal functioning of assimilating carbon. Okay. The other thing that it may be in your presentation is you're so safe with soil moisture after seconds of rain. How much of it is due to the plant functioning, and how much of it is for a given tree ecosystem? How much of it is a lag time that precipitation reach the root zone?
So let's finish with the SIF questions first. The SIF is a very direct measurement of photosynthesis because it's a byproduct of photosynthesis. So it's, for instance, something like optical leaf area index or NDVI and things like that. They basically are measuring how much shadow you're producing. This actually measures not the leaf that's there, but is it working? Okay, and in fact, but it's coarse resolution. It's subject to clouds, so it's a benchmark data set for that reason. And now they're finding that actually plants even regulate the amount of fluorescence they make depending on what type of stress they're in. So it gets complicated. Yeah. And with regards to vegetation optical depth from other frequencies, the advantage of L-band is that it penetrates, and you're not just seeing the top of the trees. In X-band and C-band, you're seeing only the top of the trees. But much more importantly, L-band is a protected channel. No one is allowed to transmit in L-band. I mean, people do, but here and there. But it's protected. So the signal that you get is pure emission from the ground. C-band and X-band, there's plenty of telecommunications
Devices,
and all that. And if you have even a walkie-talkie in a pixel that's 10 kilometers, that is such a strong signal compared to the natural signal that it corrupts it.
Any other question? Thanks, Dara. We will have another tour of Q&A. Tu as une réponse. Toi, il faut que tu payes.
I have a candid question. You were talking about stomata, and we cannot see them. You need an electronic microscope. So how do you get to manage the cycle of the openings and closings on such large scale?
We're sort of looking at the effect of them closing, or for instance, when they close, they do not fluoresce. So we measure the fact that they must be closed because they're not absorbing and re-emitting solar radiation the way they should. And also their temperature goes up, because they're not cooling by evaporating. So we look at the effects of it, not the individual. So it's,
Just to push a little bit ahead what Dara presented, is basically as he says, the carbon water cycle or carbon energy cycle are complex, and the graph that he shows that all the model, they are completely out of range. So going to the step of going to the data and analyzing it without involving models, it is one first step, because the next step is to come back to the model, because for instance, remote sensing cannot do prediction. You need to have your data in order to analyze it. But the next step, after understanding how the system works, to take all this understanding and to put it in new parameterization of the processes of interest, because we basically need to predict. We need projection, and with remote sensing alone, we don't have this possibility.
yeah. And also, the benchmark graphs that I showed, for instance, the evaporation, soil moisture, that is not directly used in the models.
Yeah.
The models have thousands of lines of code for dozens of processes, and this is the end signature. So it's sort of, it's not the individual pathways, but the aggregate behavior. So, that's the benchmark. It's like it putting night and day in the right places. You don't actually code that, it's emergent behavior.
Thank you.